In this video, we're going to be talking about base excision repair. Base excision repair is a pathway taking place inside every cell which repairs single-strand damage to DNA bases, typically damage that does not result in a large-scale disruption of the helix shape. The most common forms of damage are oxidation, alkylation, and deamination. For example, guanine can be oxidized to oxoguanine by reactive oxygen species produced as byproducts of metabolism. Oxoguanine binds to adenine instead of cytosine. This leads to a point mutation in the replicated DNA, where a CG pair is switched to an AT pair. Base excision repair uses enzymes to identify, cut, replace, and repair DNA lesions. There are two types: short patch for one nucleotide and long patch for 2 to 10. The first enzyme, DNA glycosylase, moves along the DNA strand looking for lesions, which indicate damage to bases. Note that different glycosylases are specific to different types of damage. If it finds one, like this oxidated guanine, it flips the base out of the helix. It then cuts the glycosidic bond between the base and the sugar phosphate backbone of DNA. This leaves what is known as an AP site, where there is no purine or pyrimidine at this point of the DNA strand. Another enzyme, called AP endonuclease, cleaves at this AP site. This leaves a new 3' end here and a new 5' end here. In short patch repair, DNA polymerase comes in to replace the missing bases and has an extralyase domain to cut off the extra phosphate backbone where the AP site used to be. In long patch repair, the DNA polyp performs displacing synthesis, which forms a flap of displaced DNA. This flap would be cut off by the aptly named flap endonuclease. After any new bases have been added and the DNA is now corrected, there is one final nick in the strand here. This is replaced by DNA ligase, leaving behind a fully repaired strand, good as new. This process of DNA base excision repair is incredibly important, as DNA damage can happen up to 1 million times per cell per day. If we didn't have base excision repair, these mutations could lead to numerous disorders.